and in the posterior part of the bladder, vasodiaphragm is a little enlarged. This enlargement of vasodiaphragm named ampulla of vasodiaphragm. In this point, two accessory glands empty into the vasodiaphragm. One of them named sorry, named seminal vesicle, this part. And after seminal vesicle, you can see prostate gland, this one. This prostate gland is a passageway for sending out of the body urine and semen from the out of the body. And after that, we can see third accessory gland named as bulboureteral gland. In here, the vasodiaphragm name change into the urethra. If it's inside the body, we said internal urethra, and if it's outside of the body, we said external urethra. So, my friend, testes are male gonads. They are located in the scrotum, as you see in the previous schematic figure. Testis is covered by a thick connective tissue capsule, and this capsule named tunica albungia. This capsule, this testis divided into 300 lobules by a septa. Here you are seeing lobules. These are lobules inside the testis which separate from each other by the connective septa. And inside each lobule, we have seminiferous tubules. These are named seminiferous tubules, which is a coiled tube inside each lobule. So, inside the seminiferous tubule, we can see germ cells. During formation of the embryo, the primordial germ cells migrate into the testes and become immature germ cells called spermatogonia, which lie in 2 or 3 layers of the inner surfaces of seminiferous tubules. Here you are seeing seminiferous tubules cross section. One seminiferous tubules, imagine that this is another one, this is another seminiferous tubule. And inside the seminiferous tubules, you are having spermatogonia. Spermatogonia are in the inner layer of the seminiferous tubules. You are seeing very large germ cells in the inner layer. Besides spermatogonia, we have another type of cells inside the seminiferous tubules named Sertoli cells. These are, my dear friends, these are named Sertoli cells. Sertoli cells are inside the seminiferous tubules, and there are 3 duties of the Sertoli cells in the spermatogenesis that I'm going to talk about them. Inside the seminiferous tubules, we have spermatogonia and Sertoli cells. Spermatogonia is the same germ cells or stem cells which produce finally sperm. But Sertoli cells is a kind of nerving cells or protecting cells. Sertoli cells protect spermatogonias from physical damage, chemical damage, bacteria, toxin, any kind of damages. Sertoli cells, one fact about Sertoli cells is that Sertoli cells are non-replicating. It means that Sertoli cells produce during embryonic life, a little amount of Sertoli cells produced during embryonic life, and after that Sertoli cells cannot do mitosis. It means the same Sertoli cells stay in the seminiferous tubules during lifetime of the male. There are 3 duties about Sertoli cells. The first one is production of signaling molecules and a fluid that nourishes sperm and other proteins such as androgen binding protein, abbreviated as ABP, Mullerian inhibiting substance, you know about Mullerian inhibiting substance, growth hormone, inhibin, and the aromatase enzyme. You know aromatase enzyme can convert testosterone into the estradiol. The second function of the Sertoli cells are establishment of the blood-testis barrier. Sertoli cells, as you are seeing in this schematic picture, my dear friend, Sertoli cells, this is one Sertoli cell, this is another Sertoli cell. Sertoli cells connect tightly with each other and produce a kind of barrier named blood-testis barrier. Because of tight junctions and basal compartments, they can tightly connect with each other and produce a kind of barrier between the inside the seminiferous tubule and outside the seminiferous tubule. And the last but not least function of the Sertoli cells is phagocytes of excess spermatogenesis remnants. Imagine that, my dear friend, these are spermatogonia, and spermatogonia are very large cells. So during mitosis and then meiosis, spermatogonia change its shape and produce small sperms with little cytosol, very small nucleus. So some of the excess macroorganelles or even microorganelles should be phagocytized by Sertoli cells. So this is the last function of Sertoli cells. I want to memorize all three duties of Sertoli cells: producing some agents such as androgen binding protein, melanin inhibiting substance, inhibiting growth factor, aromatase, establishment of blood testis barrier, and phagocyte excess spermatogenesis remnants. If you have any question, write in talking about each slide. I'm seeing your text box. You can ask me. I can repeat if you have any problem. So the second type of cell, or in fact the third type of cell inside the testes, named Leydig cells. My dear friend, Leydig cells are not inside the seminiferous tubules. Leydig cells named interstitial cells. It means that they are between the seminiferous tubules. Imagine that this is one seminiferous tubule, this is another one, this is the third one. So Leydig cells are in the interstitial space or in the parenchyma cells. So another name of Leydig cells in some textbooks is interstitial cells. The function of Leydig cells or interstitial cells is producing testosterone. Testosterone was produced by Leydig cells, and this testosterone initiates spermatogenesis. So here you are seeing a real histology of the seminiferous tubules. You can see spermatogonia when they are starting to produce sperm. Spermatogonia do mitosis and then meiosis 1, meiosis 2, and finally, after finishing meiosis 2, the sperm release into the lumen. This is the empty space inside the seminiferous tubule named lumen. And then this released sperm into the lumen, travel along the seminiferous tubule, and finally empty into the epididymis. So the next part that we want to discuss is about epididymis. Epididymis is the second part that sperm travel into them. It's another coiled tube. The length of the epididymis is about 6 meter. Epididymis has three regions: head, body, tail. All sperms empty into the head of epididymis and move through the head and body into the tail and going out of the epididymis from the tail of the epididymis. So most mature sperm are seen in the tail of epididymis. What is the function of epididymis? After the sperm have been in the epididymis for 18 to 24 hours, they develop the capability of motility. Even they have some inhibitory proteins that uh, inhibits the motility. It means that they can potentially be motile, but because of inhibitory proteins on the head of sperm, they cannot move. Why? Because we need to save sperm's energy. We need to save sperm is a very small molecule with a very low amount of ATP. So we need to save the sperms, so they potentially be motile. But we add inhibitory proteins on them. We have another part, sperms going out of the epididymis from the tail of epididymis and go to the vasa deferens. Vasa deferens is a straight tube contains with epididymis and enlarged to form a sac-like dilation at the posterior part of the bladder named ampulla of vasa deferens. This part, ampulla of vasa deferens. And two seminal vesicles added empty is a seminal vesicles are considered as an accessory branch. So two seminal vesicles empty into the vasa deferens in this part, in the ampulla of vasa deferens. The ampulla of vasa deferens and the seminal vesicle pass into the ejaculatory duct, leading to the body of the prostate gland and then empty into the internal urethra. Urethra transports urine from the bladder and semen when ejaculated to the outside of the body. Here you are seeing some methods for birth controlling as a contraceptive method, where some methods are considered as reversible, some of them are irreversible. In the male reproductive system, we have a irreversible method that is not very invasive, named non-scalpel vasectomy, abbreviated as NSV. They use a little opening in the posterior part of the scrotum and bring out vasa deferens by this instrument named scalpel and cut two sides of the vasa deferens and block it. So this is one surgery for irreversible method of birth controlling that is less invasive rather than female sterilization by tubal ligation. They should do laparoscopy and go to cut the fallopian tube inside the abdomen, so it's so invasive in comparison to male non-scalpel vasectomy. So, what is the meaning of spermatogenesis? What is the meaning of spermiogenesis? During puberty, the spermatogonia begins to undergo mitotic division. Here you are seeing that this is primordial germ cells that migrates into the testis during embryonic life. So spermatogonia stay until the puberty. After puberty, spermatogonia start to first mitosis, and the mitosis change spermatogonia into the primary spermatocyte. Primary spermatocyte initiates first meiosis and change into the secondary spermatocytes. Secondary spermatocytes start second meiosis and to be spermatid. Spermatid genetically is equal to the normal sperm, but as you are seeing here, spermatids are larger, circular rather than the nature sperm. And the one differentiation is happen on the spermatid to um, phenotypically change into the nature sperm. Sperm or spermatozoa, you are seeing they have head, they have mid body and tail. This is a typical sperm differentiation from a normal cell. They miss many macroorganisms and microorganisms. You are seeing that finally sperm contain head part, mid space, and tail. In the head, we have a nucleus, very compact nucleus, and acrosomal proteins. And in the mid space, we have arrangement of Spermatogenesis is started by interaction of Leydig and sertoli cells. As you are seeing here, I want you only find the important part that I'm going to tell you. This is Leydig cells, This is Sertoli cells. This is Leydig cells and this is Sertoli cells. The interaction between two cells starts spermatogenesis. How? The Leydig cells, under the effect of LH, which produce and secrete from the anterior pituitary, um, activate Leydig cells to increase amount of testosterone. My dear friend, yes, luteinizing hormone. Thank you. Luteinizing hormone, and abbreviated as LH, secretes from anterior pituitary. So, LH from anterior pituitary circulate in the blood, go to the testes. On the Leydig cells of the testes, produce testosterone. This testosterone go to the Sertoli cells. And in the Sertoli cells, we know yesterday we talked about the aromatase enzyme. In the Sertoli cells, testosterone converts into estradiol. And again, this estradiol back to the Leydig cells, and uh, the amount of testosterone will be increased. So this interaction between Sertoli and Leydig cells increase the concentration of testosterone to a much higher to initiate spermatogenesis. It means that that's right, Leydig cells involved in producing testosterone, but the concentration testosterone must be increased, must be reached to a higher level to start spermatogenesis. pKa, my dear friend, abbreviated as protein kinase A, and I don't want to memorize the second messenger or internal signaling of the each cells. Only please remember Leydig cells has LH receptors, Sertoli cells has FSH receptors, aromatase enzyme are inside the Sertoli cells, and the interaction between Sertoli and Leydig cells can increase testosterone level to start spermatogenesis. Okay? Any question regarding this slide? Okay. Here you are seeing the movement of sperm, but it's not working. So let's talk about accessory glands and male reproductive system. We have seminal vesicle, prostate, and finally bulbourethral. Seminal vesicle, my dear friend, produce approximately 60% of semen. Seminal vesicle fluid, which is the last to be ejaculated and serves to wash the sperm out of the ejaculatory duct and urethra. It has seminal vesicle has secretory epithelium that secretes a mucoid material such as fructose, citric acid, prostaglandins, and fibrinogen. Second accessory gland is prostate gland. Near to 20% of seminal vesicle is coming from prostate gland. Secretes a thin milky fluid that contains citric iron, calcium, phosphate iron, and clotting and anti-clotting, in fact, enzymes. Slightly alkaline may be quite important for successful fertilization. It means that sperms can be alive and can be more active in the neutral and alkaline medium. So, the most important gland that add alkaline feature to the semen is prostate gland. The last gland is bulbourethral gland, produce near to 5% of semen. You are seeing it's very low. So this is the precise paired and repaired based off at the base of the penis. The bulbourethral gland contributes only 5%, as I told you, and the secretion of the bulbourethral gland helps to only lubricate the distal urethra and neutralize the acidic urethra because we know urethra is a passageway for sending out the urine and also semen. So urine's pH is a little acidic, so this bulbourethral glands secretion neutralize the urethra. Here you are seeing the amount and some feature of semen. The next thing that when sperm ejaculate from the male reproductive system and go to the female reproductive system, the first step is capacitation. Capacitation making it possible for penetrate the ovary. During 1 to 10 hours, uh, some changes may be happen, but these changes name capacitation. We can categorize it as a three level. The first one, the uterine and fallopian tube fluids wash away various inhibitory protein. We know that inhibitory protein was added in the epididymis into the sperm. In female genital tract, spermatozoa gradually lose much of their excessive cholesterol on the membrane, on the head membrane especially, and is so doing the membrane very weaker. So when the membrane is going very weaker and thinner, calcium can enter into the sperm. And we know one function of calcium is starting the exocytosis. So the acrosomal proteins will be going out of the sperm by the exocytosis because of calcium. My dear friend, the capacitation can be categorized in three sessions: washing away inhibitory proteins, decreasing cholesterol in the membrane, and entering calcium. And these levels are happen by the time until when calcium enter into the sperm. The sperm are near to the ovum. So the next step after entering calcium is releasing acrosomal enzymes. So the next step after calcification is acrosomal reaction. When acrosomal reactions happen, the sperm must be near to the ovum. The sperm hyaluronidase polymerizes the hyaluronic acid and hyaluronic acid polymerase and digest the cement and the some membrane of the ovum. The prolytic enzymes digest protein in the structure of the ovum, and all of them helps to penetrate the ovum membrane. Again, this slide was a gift. You can download it if you can see it. My dear friend, I upload my files in the Navi, and also I upload files in here. You can find it the files in the files part, the MRS2. Today's topic is MRS dash 02. So let's talk a little about androgens. Androgens means any steroid hormone, including testosterone and at least five hormones from adrenal gland that generally named masculinizing hormone or masculinizing effect because the androgens are involved in increasing skeletal mass. In female, maybe we have a little amount of androgen, but this androgen seems has no effect except growth of pubic and axillary hair. So Hasnian asked me, is this file the same as? So yes. I think it's the same. Maybe one or two slides different, but I think it's the same. To be sure, you can download from here. So, what is the secretion pattern of the testosterone? During fetal life, from 7 weeks after gestation, the testes are stimulated by HCG, human chorionic gonadotropin. We will talk about this Wednesday. From the placenta to produce moderate quantities of testosterone throughout the entire period of fetal development and for 10 or more weeks after birth. At puberty, testosterone production increases rapidly, and this high level of production contains the most of their rest life. After age 50, testosterone starts to decline. For example, in age 70 or 80, the amount of testosterone being 20 percentage of normal limits. So, what is your question? I told you, please download these files in the Skype. Okay? I told you that both of them is okay, but it's better to download from here. So you are seeing the secretory pattern of testosterone in this graph as a red line. Here you are seeing before birth and after birth. In the first month after birth, the level of testosterone a little increase, but then decrease and decrease until the puberty. In the puberty, the level of testosterone increase, and also you are seeing the spermatogenesis as a sperm production and as a blue line. You are seeing after puberty, level of testosterone are in parallel with the spermatogenesis. Also, testosterone has a diurnal rhythm, and this diurnal rhythm is is obvious in the young in comparison to older ones. The proteins, testosterone should be removed from, should be bind into the some proteins to circulate in the bloodstream. So mostly testosterone bind to alpha-glucosidase or beta-glucosidase. Specific beta-glucosidase has SEG. Finally, when testosterone reaches to the tissues, in tissues, especially in prostate gland of adults and genital, external genitalia of male fetus, testosterone converts into the dihydrotestosterone. After the testosterone do its duty, the testosterone that does not become fixed to the tissue is rapidly converted mainly by the liver into androsterone, dihydroandrosterone, and is simultaneously conjugated as a glucuronide or sulfate and exerted by the urine or through the kidney. These are some secondary characteristic of testosterone. Secondary, there's some feature of the testosterone that add secondary characteristic into male. Testosterone is responsible for development of the some features such as secondary characteristic or phenotype means such as distribution of hair, baldness, basefoot, thickness of skin, protein formation, increasing amount of RBC, YBC, hematocrit, electrolytes. All of them will be increased because of testosterone. Here you are saying that testosterone can act directly or can convert into dihydrotestosterone or estrogen into some different tissues. This slide is not for the final exam. I will not ask which one testosterone in which tissue convert to which type. Only please see testosterone in the skeletal muscle, in the RBCs, in the fat, abdomen fat, or in the bone skeletons must be act directly. But in the some other tissues, testosterone can convert into dihydrotestosterone or even estradiol. About one-fifth of the amount of non-pregnant female estrogen are formed in the male. What is the role of estrogen in male? The estrogen, um, conversion of testosterone is to estradiol and then estradiol back to the Leydig cells and overproduce testosterone production for spermatogenesis. And production of estrogen from testosterone can be seen in the liver. Mostly you are seeing that 80% of male estrogen is because of producing estrogen in the liver. Yesterday we talked about the role of the testosterone on the HPG axis. We know what is the meaning of HPG axis. Can you tell me? Write it or speak it. What is the HPG axis? My dear friend, what is the meaning of HPG axis? I'm waiting. The meaning of the definition of HPG axis. The definition of HPG axis. We talked yesterday. I'm waiting, my dear friend. Hassan, what do you mean about the amount of HPG being released? HPG is not thing. It's not one composition. No, HPG is not sex hormone. It's the axis. Yesterday we talked. My dear friend, tomorrow we will have exam. I think you didn't study the files on the Navi and you are not in the class, to be honest. So on Wednesday we will have exam for the session 1 and session 2. Yesterday we talked it. Only I want the definition. What is abbreviated? And why HPG? What is the abbreviation of? Hasnian, open your mic and talk, please.
Pano, can you hear me?
Yes, yes,
Uh,
please.
Um, just so you know, it's Kuwait. My laptop isn't working, so I'm with Hassani. Is that okay?
Hassan, okay?
Thank you. So at first I didn't hear your question properly. I'm very sorry. So the HPG, it is the hypothalamus and the pituitary and the gonads. And the axis it's basically is saying that these three they work together to control the release of the sex hormones,
Excellent,
especially
thank
in
you.
females.
Thank you, Hassan. Let me write
Merci,
your
Fana.
name for extra mark for being so active. Thank you, Hassan.
Thank you, Fano.
Welcome.